Hello, welcome to Clinical Chemistry 1. I'm Rebecca Smith, your Clinical Chemistry 1 instructor. This week we're going to review chapters 1, 2, and 3 in your Bishop Clinical Chemistry book. If you read through your book, you're going to notice there's quite a bit of material and quite a few pages covered through the first 3 chapters. What I would like you to do is focus on the PowerPoints and the titles that I am going to give you in these 3 lectures that you will see in your online course. The first part, we're going to focus on a little bit of review of chemistry and some of the basic principles and practice. Much of this is review from your Intro to Clinical Laboratory Science course. Moving on to Chapter 2, we're going to look at some safety and regulations. Chapter 3, we're going to look at some of the Restigard rules and method validation. So let's go ahead and get started with Chapter 1, the chemistry review and basic principles and practice. Alright, the first thing are elements. Elements are one of the 100 or more pure substances that make up everything in the universe. Here's a picture of the periodic table that you were given in the Intro to Clinical Laboratory Science course. We don't expect you in the clinical chemistry course to memorize all of this, but we do want you to be familiar with what some of these numbers mean. Specifically, the gram molecular weight that you see underneath each element. For example, calcium is 40.078. This is what you would need to calculate something such as molarity. So just be able to use this as a tool to find the gram molecular weight when needed to create a certain type of molar or normal solution. Some examples that we just went over that are on there: carbon, oxygen, hydrogen, nitrogen, just to name a few of the elements that are on there. The ones that we refer to more in the clinical chemistry when we test for things on patients would be on this list. We do oxygen content sometimes on patients. We look at the sodium levels, their calcium levels, potassium levels, chloride levels, phosphorus levels. So you will need to be familiar with some of these when referring to the patients. Now, as we get past week 3 in clinical chemistry, we're going to start actually looking at each of the different types of tests that we do. But the first 3 weeks are just going to be a basic overview of how the clinical chemistry department works. And these are just a few elements that you may hear us mention moving forward. When we talk about an atom is the smallest particle making up elements. They consist of protons, neutrons, and electrons. Now, I don't want you to get these confused. In the clinical chemistry class, we talk about electrodes quite a bit and electrons. Make sure that you know the difference between the two. The electrons have a negative charge, and you can see them orbiting around the nucleus here. Neutrons have no charge, and protons have a positive charge. Sometimes you'll see an element that'll have a plus or a minus sign after it. If it has a plus sign, such as potassium and sodium, it means that there is an excess of protons in relationship to the other subatomic particles. If you see a negative charge in something like chloride, it means there is an excess of negative charge within there because there's extra electrons. So that's just a basic overview of why you may see a positive or negative sign after an element. So here's a drawing of carbon, and we talked about the importance of knowing the number on the bottom. The atomic mass, or we call it the gram molecular weight, is the mass of that. Okay, so we talked about Avogadro's number, 6.02 times 10 to the 23rd, and its relationship to the atom and how much would be there. So that is a review, something that is in the Intro to Clinical Laboratory Science course. I will also post my YouTubes of review of laboratory math, as we just don't have time here to review every single piece of molarity and normality. So here's another picture of a carbon atom and what it might look like. It's got 6 protons, 6 neutrons, and it has 6 electrons. 1, 2, 3, 4, 5, 6. This has a neutral charge. It would not have a positive or a negative charge. Some other things that we look at in chemistry are compounds. Compounds are two or more elements chemically combined to form a new substance. The properties are the way that the chemical substance looks and behaves. For example, here are some compounds. This one's going to be quite familiar to you. H2O, water. Two hydrogens and one oxygen form to make a water molecule. As you can imagine, water is going to behave differently than just oxygen, which is usually O2. So a compound is made of two or more different atoms to form molecules. We have two different formulas. The chemical formula, which is the H2O that you see here, which is what we see most often. And if you've ever taken a chemistry class, specifically organic chemistry, you've looked at a lot of structural formulas, which shows the arrangement of the atoms in a single molecule. And that's what it looks like here when you actually draw the two hydrogens and an oxygen. Here's an example of a molecule. This is a glucose sugar. So you can see there's a lot of energy in here, lots of carbons, lots of oxygens, lots of hydrogens. The chemical formula is obviously easier to write in something like a paper or when you're trying to put some things down in a lab report. C6H12O6 is a lot easier to draw than the structural formula. Although the structural formula is important because it shows how many binding sites each of these elements has. This is not something I'm going to expect you to do in this course. I'm just giving you an overview of what a molecule is. It's a number of things combined together. When it comes to compounds, we have inorganic compounds and organic compounds. Those of you who have taken chemistry before, especially in a four-year college type environment, you may have had an inorganic chemistry class and an organic chemistry class. The inorganic chemistry class focused on things that usually don't contain carbon. Some things might be simple table salt, which is NaCl, and water. Organic compounds are like that sugar molecule that I just showed you, very complex. So here are some of those examples of inorganic compounds I was talking about. Water, hydrochloric acid, carbon dioxide, and your typical table salt, sodium chloride. Okay, not a lot of carbons there. Then we have the organic compounds. This is where we have a lot of elements making very large compounds. These may sound familiar when it comes to your diet: carbohydrates, lipids, and proteins. Those are organic compounds. Okay, so that's how you can recognize those go in that category. They're very large and they have a lot of elements and a lot of carbon in them. Another one down the bottom here, nucleic acids as well, is what DNA and RNA are consisting of. So we're going to go back and do a little bit more review quick of the Intro to Clinical Laboratory Science course. When we look at the different units of measure, we use the SI system. It's based on the metric system, and I'm not going to review the metric rule. You guys did a lot of that in Intro to Clinical Lab Science. But we look at the basic units based on length, mass, or quantity of a substance. Length would be something like meters. Mass would be something like grams, for example. We also look at different chemical grades of purity. Different varying grades might be analytical reagent grade, ultra pure, chemically pure, United States Pharmacopoeia, and National Formulary. The American Chemical Society established specifications for analytical reagent grade, or AR chemicals, that manufacturer will meet or exceed it. These are suitable for use in most analytical lab procedures. We typically use reagent grade types of items. You can get them extremely pure and they get more expensive. And you can go down to things that you'd find at Fleet Farm or Home Depot that you take that oil off of your pavement, like muriatic acid. That would not be something that would have a high grade of purity. Okay? Those types of things that you find there for $1.99 a gallon. But you can get up to things that may cost hundreds of dollars a gallon, which are ultra pure and have no impurities in them whatsoever. Here's an example of a reagent. Reagents are things that we use in clinical chemistry to drive a reaction to happen. We add the patient's sample to a reagent, a certain type of chemical reaction happens, and we monitor that reaction by way of a color, a light produced, some type of energy is produced from that reaction, and we have a way to measure it. So this picture shows an example of many reagents for all different types of tests that you might find on a clinical chemistry analyzer. Then we have standards. We need to make sure that our clinical chemistry analyzers are working appropriately. So we use something called a standard that we purchase from a manufacturer, and we know that this chemical is measured to produce an exact known concentration and purity. They are NIST certified, which is a standard reference material, and it is proven that if they're certified by NIST that we can know with complete certainty that it has that exact amount in it. So for example, if we just had an analyzer, we turn it on and we run patient samples for glucose on it, we need to be able to tell that analyzer exactly how to know what certain levels of glucose look like. So that's what our standards are for. We run standards on there. Let's say a standard of 100, a standard of 150, a standard of 200, so that when you put your patient on there, on the analyzer, and run it, it compares that answer that it gets with those standards and derives its answer from that. These standards will make a lot more sense to you as we move through the clinical chemistry course and you use them. Okay, so if you're kind of leery in the difference between quality control and a standard, it's okay. I promise you by the end of week 4 and 5 you will completely understand because you will be actually using them. With water specifications, it's important to know where each type of water comes from. Tap water is usually unsuitable for laboratory procedures. Sometimes if you take a drink of water from like a drinking fountain or something like that, it tastes like copper, it tastes like chlorine. It's okay, I mean we drink it and it's just fine, but when it comes to our chemistry analyzers and things that are so particular, we have to make sure that we don't have any types of impurities that can create improper lab results or inaccurate lab results for us. So what we do is we purify it in different types of ways. The first one, distilled water, is purified by distillation. Here's an example of a modern day distiller. When I was in college, we would distill using a Bunsen burner, and we had a glass bulb, and the water went up to the top and it dripped down. This works the same, but it uses electricity, which is probably a little bit easier. What happens is you have a bunch of water on your right hand side here, and it's heated. As it heated, a water vapor is created. It comes through this coil here, and it's cold. Okay, so as it cools, the water comes down the bottom. All the impurities are left in the bottom of the hot water. Those don't go up in the air; they stay there. So you're going to end up with, after you clean this thing out, probably like a crust on the bottom. So you do have water that has less impurities in it. Then we have deionized water. Distilled water may still have some positive and negatively charged ions inside that liquid. That can still interfere with some of our electrolyte testing or testing for sodium, chloride, potassium, and things like that in our chemistry analyzer, so we want to make sure that those items have been removed. So deionized water is purified by ion exchange. It uses other positive or negatively charged items to pull those out of the water. Reverse osmosis water is something you may have heard of. A lot of people that live out in the country sometimes have these installed in their homes. It pumps water across a semi-permeable membrane. If any of you have taken the biology class and you've taken the dialysis tubing and you put the pancake syrup in it and you put it in water, you noticed how it moved across that membrane. Reverse osmosis works very similar to that. And if you have not done that, you will be doing it in the biology course. At least I think they still do it. We usually use reagent grade water, and we purchase water quite a bit, or some labs that go through a lot of water may actually make their own, but the smaller ones definitely do purchase. We call them cubitainers of water. They usually have about 3 to 5 gallons in them. Type 1 is the most pure. It has minimal interference. It doesn't have trace metals or doesn't interfere with trace metals or enzymes. We use it for quantitative and qualitative procedures, so know that type 1 water is the most pure. Type 2 is acceptable for most analysis, although type 1 is the of choice. And type 3 is acceptable for glassware washing, but we wouldn't use it to make our reagents or anything. Not only do we use specific types of water, we have to make sure that water is safe. We do colony counts where we send it to the microbiology department to make sure bacteria isn't growing in our water containers. We need to watch the pH. Too high a pH or too low a pH can mean there's too much acid or too much base inside of our water. And we look for levels of chlorine, ammonia, iron, hardness, phosphate, and sodium. So obviously if we're purchasing type 1 water, we would not have those items present. But if you're using some type of distilled water or whatever, you may check for those types of levels. This will bring you back to high school or middle school, colligative properties. The ones that you're probably familiar with are the second, third, and fourth ones here. Vapor pressure, which is where liquid becomes a gas. Freezing point, where liquid becomes a solid. And boiling point, where water becomes vapor. So the one that's going to be very huge in the Clinical Chemistry 1 and 2 course is osmotic pressure. This is the pressure that allows a solvent to flow through a semipermeable membrane to establish an equilibrium. Now, with this, we look at a lot of things in the human body, and it's very important to make sure osmotic pressure is maintained, and your body does a really good job at this. Through your brain barrier, you always want to make sure there isn't a too high or too low osmotic pressure there. Your cells, you want to make sure there isn't more pressure on the outside of the cell than there is on the inside of the cell. So there's many different types of things in your body that are going to help control this osmotic pressure. We call it osmolality and osmolarity when we get to that section at the end of Chemistry 1. But that pressure across that semipermeable membrane is so important in the human body that we're going to spend a lot of time going over how that works and how our body controls that. Very exciting. Next thing is redox potential. This is where we talk about our positive and negatively charged atoms and what they do. The redox potential measures the ability of a solution to accept or donate electrons. If it accepts electrons, it's known as an oxidizing agent. If it donates electrons, it's known as a reducing agent. You don't have to go into a lot of detail on this. Just understand that redox potential causes the donating or accepting of electrons. And when we see how later on how things are working within a reaction in clinical chemistry, you'll see more of this. pH and buffers are important. Know the definition of a buffer. This is a weak acid or base and their related salts which minimize the change in hydrogen ion concentration. In general, it keeps the pH where you want it. Buffer keeps the pH where you want it. Okay? If this definition bothers you, if you know that it just keeps it where it needs to be, you're good. Here's an example. We do enzyme testing in the clinical chemistry laboratory. Enzymes are so picky, okay? They will change in just a small budge of the pH. If you clean out your glassware and you don't rinse it well enough and there's a little something left in there and it throws the pH off, your whole chemistry reaction won't work. By putting a buffer in the reagent, it makes it so that pH cannot change even if there were something that we're trying to change it. So it resists a change in pH. Okay? Very important. We really like to have buffers in clinical chemistry because it keeps our sanity. Keeps the pH from changing. You don't want it, you want a 7.4 or whatever. You don't want a 7.6. So it'll keep it there for you by adding that buffer to it. pH is usually on a scale of 0 to 14. A low pH of less than 7 is usually acid. Around 7 is neutral, and greater than 7 is usually a base. We talk about the Henderson-Hasselbalch equation. I don't make you memorize it. I just want you to be associated with it. But it is the equation here that we use to calculate pH. It takes into account the amount of bicarb, which is the top HCO3, and the amount of CO2 acid, which is on the bottom, plugs it into a calculation, and that's how we get the pH. So just know Henderson-Hasselbalch equation is used to calculate the pH. I won't be testing you on this. You'll have it on a worksheet in the future, but you'll just have to use this calculation with your calculator. As long as you have a log button on your calculator, you'll be able to figure it out. The bicarb would be given to you, the CO2 would be given to you, and the log button. So 6.1 plus the log of the bicarb over the CO2 would give you a pH. So the normal ratio of bicarb to CO2 is 20 to 1. So this would be a normal pH right here. All right, let's look at some pipettes. We used volumetric pipettes and serological pipettes in the Intro to Clinical Lab Science course. There was a couple different types that were talked about. A to contain holds a particular volume but doesn't dispense that volume. A to deliver, which is what we mainly have in our laboratories, dispense the volume indicated. And if you see the etched rings on the top, it means that it is a blowout pipette. If you do not see the etched markings, you just let it drain. More pipette does not have graduations to the tip. It's also self-draining. A serological pipette has marks to the tip and is usually a blowout pipette. We also have Pasteur pipettes, which are just the transfer pipettes that don't have any markings at all. I'm going to show you a little video here of review of pipetting technique. So he's going to show you how to draw air into the pipette continuously as you insert it into the solution. What I like to do at this point is push my pipette down to the bottom as he does here to prevent it from draining out too quickly. Make sure that you use your dominant hand to hold the pipette with your finger close to the top. You're going to draw with your non-dominant hand with the bulb so you have more control. I've seen quite a few different types of techniques in my Intro to Clinical Lab Science course, but this is the technique that we want you to follow. Some people say, well, what difference does it make what hand you use? Why can't you use your thumb on the top? It does make a difference because when you go out in the clinical laboratory world, this is how everybody's going to do it. So you want to make sure that your finger of your dominant hand is what you are using and the bulb is in your non-dominant hand. Okay, next review topic is centrifugation. This is how we separate the solid from the liquid. Some other things that you need to know here with phlebotomy, you're going to learn it in your phlebotomy course. You're going to learn the different types of tubes, which most of you are taking right now in conjunction with Chemistry 1. So here's some important tips that you need to know. First of all, balance your centrifuge. You've learned that. You had to do it on your intro final. You also want to use red top tubes have to sit and clot for 30 minutes before you centrifuge them. Green top tubes are used for stat testing. They don't have to clot. Actually, they will never clot. We need to follow safety, balance it, don't open it while it's spinning, and make sure you clean any spills right away. And it is very important that the serum is removed from the cells within 2 hours or chemistry results will be affected. So after you centrifuge, make sure you take that plasma or serum off the top of the cells. Those cells are hungry, even though they're not in the human body, and they're going to start eating up glucose that is inside that serum. So it's going to drop the glucose level dramatically. Dilutions. Dilutions are the ratio of concentrated material to a total volume of solution. We did a lot of these towards the end of Intro to Clinical Lab Science, and we're going to review a little bit more with these because we're actually going to make dilutions in the clinical chemistry course. A dilution is always less concentrated than the original, and the dilution factor will increase as the concentration decreases. I'll have an example of a serial dilution coming up which will show that. So for example, if you need 100 mils of a 1:4 dilution, you would take 24 mils of a concentrated solution and 75 mils of a diluent such as water. Your total parts is 4. Okay, so 1:4, 4 would be your 100 mils and 25 is your 1. Okay, so total of 100 and 25 of it would be your concentrated solution. Here's an example that helps a lot of people too. Here's a 1:10 dilution. Okay, there's 1 drop of patient serum and 9 drops of diluent for a total of 10 parts. This is a 1:10. I have so many students get this confused. Make sure that you know 1 mil added to 1 drop added to 9 drops is a total of 10 drops. That's a 1 to 10. You could also have 2 mils added to 18 mils would also be a 1 to 10. Here's the serial dilution that I was talking about in the prior slide. We've done these in class and we'll do them again. We'll do them again in immunology as well. But you have multiple progressive dilutions. An example would be dilute a 1 to 2 and then a 1 to 4 to a 1 to 8. And on the picture I have down on the bottom, we have the whole, we'll pretend this is patient sample. The whole patient sample, and then we dilute it in half, we dilute it in half again, we dilute it in half again, we dilute it in half again. So we end up with a 1 to 2, a 1 to 4, a 1 to 8, 1 to 16, 1 to 32. And that is how our serial dilutions work. And don't worry, you'll be doing more of those. Some things that we need to consider with our specimens that are being drawn in the laboratory is how are they collected? A venipuncture, capillary collection, arterial collection. Is it plasma or serum? And we also do things like whole blood, urine, sweat, saliva, spinal fluid, synovial fluid, amniotic fluid, pleural fluid, pericardial fluid, and peritoneal fluid. Some of the ones on here that you may not be familiar with, synovial comes from your joints, okay, so it could be elbows and knees and things like that. Amniotic is the sac where the baby is. Pleural would be something with your lungs. Pericardial is around your heart. Peritoneal would be what your guts are floating in, or your stomach and your intestines and such. Things that we see for samples would be venous blood. Sometimes it's centrifuged. Once it is centrifuged, it is either serum or plasma. You're going to learn more about how we get these types of samples in the phlebotomy course, but serum is what we have after we let whole blood clot and then centrifuge it. There is no clotting factors in serum. Plasma has an anticoagulant in the tube, so it would be like a green top tube, stays liquid all the time. When we centrifuge it, the plasma still has those clotting factors in the plasma. Some chemistry tests that just doesn't matter. Some of them it does, and we'll learn what those are as we move forward. Factors that affect blood collection, we have to look at these. You're going to have a chemistry assignment that's a little bit challenging, week 1, and it's going to talk about some of these types of things. Some of the factors are anticoagulants. Okay, so you might need to use certain tubes with anticoagulants. You might need to have certain tubes with preservatives in it. It's going to matter where you collected it. Is it a finger stick? Is it an arterial puncture? Is it a venous puncture? Is it hemolyzed? Hemolysis is when the cells are broken due to a traumatic draw. Okay, so if you're drawing from a little vein and those poor little blood cells are getting popped, all of the potassium inside the cells are leached out into the plasma or serum, and it messes with our results quite drastically. So I'm going to give you a quick run through each of these types of tubes that you might see. You're going to get these again in the phlebotomy course, so this isn't going to be the only time you ever see it. But heparin tubes are used most often in clinical chemistry because we don't have to wait for them to clot. You can centrifuge them right away. They sometimes have sodium, potassium, lithium, or ammonium salts in it. The most common one we use is called lithium heparin. Okay, lithium heparin tube. It's in a green top. We don't have to wait for it to clot. It is great for stat testing. Next one is EDTA, otherwise known as ethylene diamine tetraacetic acid. It binds up all the calcium in the tube so it doesn't coagulate, so it does not clot. It stays liquid. This is not suitable for calcium and iron specimens, as you can imagine, because it binds up all of that, or chelates it. Chelate means binding. We can use it for glycated hemoglobin samples or any type of hematology sample where we want a whole blood liquid sample. They're usually a pretty little purple top like this. Sodium fluoride is not a great anticoagulant, but it's good for stopping the cells from eating glucose. It prevents that from happening. So therefore the glucose can be maintained for a long period of time. Usually a gray top type tube. Also used on an lactic acid. Oxalates, sodium, potassium, ammonium, and lithium salts are combined with oxalic acid. This inhibits several enzymes and are used for certain types of testing. A little more rare. Only time I've ever used those was for drunk drivers. They drew them for alcohol, so the alcohol content would maintain the same in the tube until it got to the state lab for testing. Here are the red top tubes I talked about. They're usually glass and don't have any additives. There can be plastic and have a clotting activator to make them clot faster. We use these when serum is required. Sometimes we call them red top tubes, and sometimes they're called serum separator tubes if they have a plasma in the, a gel in the middle, silica type of gel. They have to clot for 30 minutes before we centrifuge them, which is why it's not ideal. Most doctors don't want to wait around an extra 30 minutes if they don't have to. With everything in these types of tubes, you can see a whole rainbow of colors here, and you'll understand what every single one of these means by the end of your phlebotomy course. There's a certain order of draw. These big tubes on the end here are blood culture bottles. This other one here, this yellow one, is also a type of a blood culture bottle. Then we would draw a navy blue top tube, which is meant for trace elements, to light blue top red. I don't expect you to know the order of draw for my course. Just know there is an order of draw, and it's very important because the tubes down here could contaminate the tubes over here. So you want to make sure that as you're filling these tubes, you fill them in this order. I talked about hemolysis earlier, and that's when those red cells get destroyed by way of a really tough blood draw. And this is what it looks like. The one on the left-hand side is a normal serum. It's usually kind of yellow. And you can see that as those cells were chopped up through whatever mechanism caused it, those cells leached the potassium into that plasma, and it's going to really interfere with your potassium, magnesium, and phosphate levels. Some of the sample processing you need to be familiar with. Proper identification is very important. When the nurse draws or the phlebotomist draws at the patient bedside, they make sure that they ask the patient if they're capable or check the armband if they're incapable. That identification stays with that tube. When it gets to the lab, you have to be very meticulous at making sure if you have to aliquot a little bit of plasma from one tube to another that it's labeled correctly. Plasma and serum need to be separated from the cells within the 2 hours, which I mentioned prior. And procedures should be followed for proper preservation and storage. If a reference lab, say Mayo Clinic, wants a frozen specimen, you need to send it frozen or they will reject it. There are some things that are going to affect certain tests. This will be part of one of your written assignments coming up as well. Pre-analytical means before analysis. Some things that we control are physiological, your diet, lifestyle, and any drugs that you may be taking. So those things can affect your blood results. For example, if you eat a big cheeseburger before you go in for your cholesterol triglyceride testing, it will affect it. Things that you can't control: biological influences, environmental factors, long-term cyclical changes like when you get older, and underlying medical conditions. Those are things that could affect your sample. And we just went through some of these. The phases of testing: the pre-analytical, analytical, post-analytical. This is not new to you. This is review. Pre-analytical is before the specimen reaches the lab. Analytical is everything in the testing. And post-analytical, everything after the testing process. And the last thing that I have in this section is a chain of custody. These are tests linked to a crime or an accident that may require tamper-proof tape and document receipt of the sample. An example is I used to work at a large hospital in Green Bay, Wisconsin, and I worked the night shift, so we got a lot of drunk drivers. Of course, we're a big Packer town, so we get a lot of people going out after the games and such. So I would bring in quite a few of these a night, and we would have to draw a legal blood alcohol. With that legal blood alcohol, I had to sign a form and sign the tube. I had to put it in a box and add crime tape around the outside of it. It would then go to the state lab. The person that opens it then takes that sheet of paper out and they sign it. They do the testing. So it's a chain of custody. Everybody that's ever touched that sample has to document that they have done so. All right, this concludes chapter 1, one of the longer chapters. Please move forward and watch the chapter 2 and chapter 3 videos next.